Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh and a very good morning. A warm welcome to the IMR-SEAMEO webinar series 2022 to all participants from Malaysia, the SEAMEO network and affiliated countries. For info, this will be the last webinar for the year. I am Dr. Mariana Ahmad, Head of the Acarology Unit from the Infectious Diseases Research Center in the Institute for Medical Research, Malaysia, and will be the moderator for today's webinar. For those who are not familiar, Acarology is the branch of science which deals with the study of ticks and mites. The research focus of the unit is towards any ticks and mites of public health importance. Currently, our focus is mainly on ACARI-borne diseases involving detection of bacterial pathogens such as the spotted fever group Rickettsia, Borrelia, Anaplasma, and Ehrlichia. Apart from that, the Acarology unit is also working on the development of point-of-care diagnostic assays, which include LAMP, aptamer, and real-time PCR assay for screening of pathogens in ACARI and their animal hosts. Ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on biomedical research, and present research outcome. Before we begin our presentation, allow me to announce a few housekeeping messages from the Secretariat. First, we will not be taking any questions during the presentation, but participants may post their question using the link that have been shared in the YouTube channel. Second, the question and answer session will be conducted at the end of the second presentation. The attendance link is also provided for those who would like to receive an e-certificate of attendance. You may find the link in the YouTube channel. Fourth, please subscribe to our National Institutes of Health Malaysia YouTube channel and click for notifications whenever there is new post. Ladies and gentlemen, the theme of our webinar today is ACARI, tiny creatures with significant health risks. For today's webinar, we have two speakers from the unit of Acarology IMR Malaysia. Our first speaker is Puan Ernieenor Faraliana binti Ceklah, who is a research officer with 14 years of experience and is currently doing her PhD in genetics in Universiti Kebangsaan Malaysia. Upon joining IMR, her research areas of interest are mainly towards control of dust mites, and then slowly shifted to ticks and tick-borne diseases. She also involved in screening of trigger mite for scrub typhus for diagnostics. She has published many papers in international peer-reviewed journals. For today, Puan Ernie will be sharing some of her research experience in the presentation entitled Ticks in Malaysia, the Facts and the Fears. Puan Ernie, the screen is yours.
Thank you, Dr. Mariana, for your kind introduction. Assalamualaikum, selamat sejahtera and very good morning to everyone. I'm Ernieenor Faraliana Ceklah, Research Officer from Acarology Unit, and my today's presentation is on ticks in Malaysia, the facts and the fears. Please allow me to introduce again the ticks in general. Ticks belong to class Arachnida and subclass Acari. There are three families of ticks, however, two of which have a medical significance, which is Argacidae, soft ticks, and Ixodidae, hard ticks. Ixodidae ticks receive more attention in research due to highest transmission rate of disease and disease-causing agent. Ticks are different from insect by the absence of antenna, no wings, and body not segmented. They are blood-sucking arthropod which widely present in tropical and subtropical region. Presently, ticks are parasitized mammals, birds, reptiles, and also human as their accidental hosts. Throughout their life cycle, all active stages of ticks feed on blood to complete their life cycle. Most of the tick-borne pathogens are transmitted by hard ticks, and they serve as a competent and highly efficient vectors due to several factors. Number one, hard ticks consist of large blood meal capacity, which means that adult female can suck blood up to 2 ml per day, and with no interruption, they have a long duration of feeding. Hard ticks present at high abundance in the environment because female ticks can lay 1,000 eggs, thus produce 1,000 offspring in their lifetime. Third one, ticks can transmit the pathogen either by transovarial or transstadial transmission. Transovarial means the infected female passing the pathogen to their eggs and thus to their succeeding generation, while transstadial transmission occur when pathogens remain from infected larva to their next life stages. In Malaysia, there are 47 tick species have been recorded, belongs to 7 genera. The most abundant tick genus was Haemaphysalis, which consists almost 20 species. Almost 2/3 of Malaysian ticks have been recorded to bite human. These are technique how we collect the ticks in the field. The first technique is animal trapping using wire trap. From this technique, we focus on trapping of small mammals, which is rodents and shrew. And from this technique, we collect on host ticks, and they are either partially fed or fully fed. Second technique was vegetation, where by searching the ticks on the lower vegetation along the trail, and if the ticks present, we collect the tick by the direct hand picking. The third technique is flagging by sweeping lower vegetation using white cloth and collect the ticks. Vegetation and flagging techniques, we collect the questing ticks, a stage where ticks are crawling up to the lower vegetation and seeking to get closer its potential host. Field studies of ticks should be based on the correct identification of the tick species collected. According to CDC 2022, it is important to identify the tick species accurately since specific tick species carry different pathogen, thus cause different infection and require different drug treatment. In Acarology unit, we integrate both phenotypic and genotypic identification of local ticks. However, phenotypic identification based on morphological characters can be difficult if the body of the ticks are incomplete, broken, not intact, and also if the tick sample are engorged with blood where some of the morphological characters are changed. For species complex such as Rhiphicephalus, there are close similarity across different species, and incomplete existent taxonomic keys, especially in larvae and nymphs, has made phenotypic identification become less applicable. To overcome the difficulties in phenotypic identification, we conduct a study on genotypic identification using DNA sequence to verify the morphological status and at the same time to produce a DNA barcoding for local ticks. To date, we have successfully produced and deposited several DNA sequence of local ticks based on the CO1 and 16S gene. And these are two new recent findings that have been published in peer-reviewed journal related to phenotypic and genotypic identification of local ticks. In the first study, 5 species of questing dermacenter ticks were collected from active recreational area in Pahang and Terengganu. This study produce useful information on the species richness of dermacented ticks, which potentially associated with tick-borne disease and might be circulated in our human population. In the second study, Amblyomma jomaidae ticks, a ticks that previously known as a tortoise ticks, has found to be infested on tree shrew, a Tupaia glis, and this finding produced the first published report of Amblyomma jomaidae parasitizing Tupaia glis, thus produce new tick-host association record in Malaysia. The infestation of Amblyomma jomaidae on Tupaia glis is considered a risk, since these tick species are considered as a vector for Borrelia and feeding on human. Presence of Tupaia glis, especially in human settlement and fruit orchard, give the risk for human to get the infection. Why ticks are important? The first, as a vector, ticks can transmit multiple pathogens, namely protozoa, rickettsia, spirochete, and viruses. A single tick can transmit multiple pathogens simultaneously in a single blood meal, thus can produce multiple infections. Tick-borne disease in Southeast Asia are mainly due to Borrelia, Rickettsia, Anaplasma, and Ehrlichia species. On 2017, Ministry of Health Malaysia under Disease Control Division has declared that tick typhus as one of the notifiable arthropod-borne disease. And these are three genera of ticks that have public health concern in Malaysia. The first genera was Ixodes, with Ixodes granulatus are known as a potential vector for langat virus, Lyme borreliosis, and rickettsiosis. Second genera was Dermacenter. As we can see that different species of Dermacenter transmit different pathogen, thus cause different disease. The third genera was Haemaphysalis, with Haemaphysalis hydrasis serve as a vector for Lyme borreliosis and spot fever group rickettsiosis. To prove that our local ticks serve as a potential vector for tick-borne disease, we conduct a study on the presence of Borrelia in on-host ticks that have been collected from several ecosystems in Pahang. Throughout the study, 2.6% of ticks from one species, which is Ixodes granulatus, were infected with Borrelia miyamotoi, a pathogen that can cause a relapsing fever, while 7.8% of ticks from two species were positive with Borrelia wallaceiana, a pathogen that associated with Lyme disease. This finding produced the first molecular report of Borrelia miyamotoi detected in our local Ixodes granulatus ticks. Early this year, we conduct a next generation sequencing analysis to characterize the bacterial community that associated with on-host Ixodes granulatus collected from 3 active recreational areas in Pahang and Terengganu. From the analysis, a total of 15 phylum and 100 bacteria genera were detected, and our local Ixodes granulatus harbor 4 main tick-borne pathogens, namely Rickettsia, Borrelia, Borrelia, and Ehrlichia. And interestingly, there is co-infection of Borrelia and Ehrlichia was reported for the first time in Ixodes granulatus. Thus, it will pose potential health concern in the context of co-transmission to human, especially in the high population areas with Ixodes granulatus. The co-infection between pathogens may lead to increased disease severity, as it will complicate the diagnosis and treatment. Besides transmitting pathogen, ticks also can cause a nuisance if they present at high number, especially at home or workplace. And most of the nuisance cases are related to the dog ticks, which is a Rhipicephalus species. As I mentioned before, human can serve as an accidental host for ticks, and early this year we received nuisance cases that caused by soft ticks, and after investigation the suspected host, which is bats, we found bats that roosting inside the house. Then after we collect the fresh guano, 2 individuals of soft ticks, probably from Argus species, was discovered in the ceiling of the residing house. Humans serve as accidental hosts for ticks, and these are 3 examples where ticks were found on human body, and the tick sample was sent to Acarology unit for the species identification. Ticks also important because it can cause human autoacarisis, an incidence where ticks were invading or infesting on human ear canal, and this incidence was common especially in the rural area because they have close contact with the ticks' habitats and also presence of pets inside their house. In a serious cases, long attachment of ticks inside ear canal can cause facial paralysis due to irritation of the nerve with the release of neurotoxin that present in tick saliva. The incidence of human paralysis have been reported worldwide, including in Malaysia, where the human paralysis cases have been reported in Pahang and Kelantan. And these are few case reports of human autoacarisis reported in Malaysia. The first case is from Kelantan. The person might infested with ticks while playing near the bushes area surrounding their house, and they go to the hospital for ticks removal, and the medical officer successfully remove the ticks, and as we can see in the picture, the ticks is black in color, which means engorged with blood. In the second case, the infestation might occur from their pets, and surprisingly, this kid was infested with ticks in both ears. And this is a recent finding that reported few days ago in Kedah, where one boy were infested with ticks inside the ear canal. According to the complainant, they said the infestation might come from their pets, and their house also close to the ticks habitat, which is a shrub. To remove the ticks, they used a traditional methods, which is tobacco extract, just to weaken and probably disrupt the tick breathing before detach the ticks from the ear. Besides plant extract, other chemical that can be used to remove ticks is Vaseline, petroleum jelly, glycerin, and lidocaine. But please bear in mind that ticks removal using chemical or plant extract is only possible for newly attached ticks and before the semen-like substance was formed around the mouthpart, because the semen was functioned to grip the host skin strongly. If that happened, the mechanical ticks removal must be applied to the patient. If you go for outdoor activities such as hiking or camping, these are little awareness and few tips that can go a long way to keep you safe from a tick's bite. Number 1, please do your tick check after your outdoor activity, especially in hairs, armpits, and also behind the knees. Apply the repellents such as repellent that contain DEETs or permethrin to keep ticks away from your surrounding. Please wear light color, long sleeve shirt and pants to avoid the bites from ticks. If you are being bitten by ticks, remove the ticks as soon as possible to avoid the risk of any infection. If you have pets in your house, regularly check your pets when they come from outside. And if you are being bitten by ticks and later develop tick bite symptoms, please go to the hospital for further treatment. As a conclusion, please be aware of the presence of ticks in your surrounding, because this tiny ectoparasite can cause a big problem in terms of disease transmission and nuisance. Ticks and tick-borne disease are rarely capture national attention in Malaysia. However, there is no room for complacency as the tick population is growing due to deforestation and global climate change. Thus, tick-borne disease become a public health threat nowadays. Based on the lab finding, more recent potential tick-borne disease, especially among rural population, have been reported. Thus, we in the Acarology unit will continue to study on ticks in order to know the real burden of tick-borne disease and at the same time to provide evidence-based finding to our stakeholders for proper control measures. Before I end my presentation, I would like to thank Ministry of Health Malaysia, IMR and NIH for their approval to conduct a tick study. Our gratitude is also extended to Forestry Department of Peninsular Malaysia, State Health Department, Science Officer Entomologists from Ministry of Health Malaysia for their assistance in the tick sampling. Last but not least, thank you to staff of Acarology unit for their direct and indirect contribution towards the tick study in Malaysia. With that, I end my presentation. Thank you.
Thank you, Puan Ernie, for the interesting talk. If there is any questions that you would like to ask about her presentation, kindly submit your question using the link provided in the YouTube channel. Next, I would like to introduce our second speaker, Puan Suhaili Zainal Abidin. She has been in the service for 18 years and upon joining IMR, her research interests are mainly towards control of dust mites and then slowly into chiggers and scrub typhus. For diagnostic service, she involves in the identification of mites, specifically scabies mite and other mites causing nuisance to human. It is our great privilege to have Puan Suhaili with us to share her experience in the presentation entitled Vector of Scrub Typhus in Malaysia, What Should We Know? Over to you, Puan Suhaili, and the floor is yours.
Thank you so much, Dr. Marina, for your kind introduction. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, and a very good morning to all the audience. It is a pleasure to be here. I am Suhaili Zainal Abidin, researcher in Acarology unit IMR. My presentation today particularly is about vector of scrub typhus in Malaysia. What should we know? Let me start briefly about what is scrub typhus diseases. Scrub typhus is a febrile illness or fever caused by the intracellular bacterium Ornithia susugamushi. Scrub typhus is one of the oldest recognized vector-transmitted diseases, and then first isolated and identified by the researcher in Japan in 1930. This scrub typhus transmitted to human through the bite of infected larval stage trombiculid mite called chiggers. The World Health Organization (WHO) has named scrub typhus as one of the most misdiagnosed and underreported diseases that often requires hospitalization. Scrub typhus is endemic across extensive part of Asia, Southeast Asia, Australia, and the Pacific region. These endemic regions called as Sugamushi Triangle. Approximately 5 to 14 days after being bitten by the infected chigger, patient will begin to have symptoms such as fever, non-specific flu-like symptoms, rash, and the most important, scar at the bite site. Laboratory tests become mandatory for the confirmation of the diagnosis when scar are absent. Available diagnostic tests are available in the clinic and laboratory, And they have two types of diagnostic tests. The first one, direct method, which is using PCR isolation of cell culture. And then indirect method, using the agglutinate of antibodies, such as well-Philic test and so on. And we move to the main vector of the scrap typhus. Orinchia susugamushi is transmitted to mammalian host human through bites of infected mites. Chiggers actually act as a primary reservoir for Orinchia susugamushi and remain infected through their life cycle. In real nature, they have two populations of chiggers, either infected or non-infected. And then this Orinchia susugamushi can be transmitted via transovarial and transstadial. And what is chigger? Chigger is a parasitic larval stage with a 3 pairs of legs of prostigmatid mites that belong to the family Trombiculid in Acari. Chiggers is really small, with a size only 0.15 and to 0.25 millimeter long. Chiggers actually attract to the carbon dioxide released by the human or the animal host, and chiggers will be attacking rodents and other small mammals for feeding. Another info for chiggers do not suck blood. Chiggers will feed on tissue lysates or fluids in skin cells of hosts. Chigger will be penetrate the host skins and inject a digestive enzyme that breaks down the digest the skin cells. The allergic reaction to the chigger saliva throughout the feeding time develop a formation like a hardened tube, and this hardened tube called stylostome. Chiggers will feeding at last for 2 to 4 days. Life cycle of the chigger will take about 59 to 135 days. After larvae molt from the jutoworm, the unfed larvae normally will be climb into the tips of the grass or stalks and waiting to clinging at the host, or maybe human pass by them or resting near the area. Chiggers in Malaysia can be classified into 7 ecology groups based on color of unfed chiggers, either white, yellow or orange. The second one associated with habitat, either in the grass, oil plantation, coastal area and so on. And then the third one will be based on association with feathered hosts, such as rodent, shrew, bats, birds, even amphibian. Normally, Leptotrombidium deliense will be in orange and yellow in colors. This is the among chiggers have been recorded in Malaysia, and then this is the chiggers actually abundantly found in small mammals. Leptotrombidium deliense have been identified one of the most vector to the scrap typhus diseases since 1930s. Chiggers in the genus Leptotrombidium are known to be a vector of Orinchia susugamushi, primarily feed on the small mammals. In Malaysia, we have already identified 3 species as a vector of scrap typhus. The first one is Leptotrombidium deliense. This species of chiggers can be found in forest and oil palm plantation. The second one is Leptotrombidium Ernieenor. These chiggers easily can found with a high distribution at the coastal area. And the third one is Leptotrombidium fletcheri. These chiggers normally can found at the wheat fields. We can avoid chigger bites by taking certain precaution when spending time outdoors. Before we go to the outdoors activity, we can wear long pants, long sleeve shirts, and bright colors of the outfit. Avoid spots where chiggers are commonly found. Use a bug repellent or insecticide as a barrier. And then we also can do discourage of the rodents near our surroundings. And once we come back from the outdoors activity, we can take a hot shower at the moment that we have realized that we have been bitten by the chigger. And then we also can look at the sign of the chiggers, and then please clean the area of the bitten, avoid scratching on the bites of the chiggers, and then try to apply the topical cream to relieve the itching. And must remember, we must wash our clothes with a hot and soapy water. Sorry. This is the flow of the scrub typhus research in Malaysia. Actually, the scrub typhus diseases have been started as early 1914 and focus more on causes, transmission, treatment, and prevention. And the study was shifted to the ecology, distribution, and zero prevalence of the scrub typhus in general of Malaysian population. For the past 10 years, various studies have been conducted by researchers in Malaysia. For example, in 2010, unit Acarology have been conducted a laboratory evaluation of 4 commercial repellents against level Leptotromydian deliense, and then by year, actually the research have been shifted more to the molecular on detection of the Orangia susugamushi in both host and the vector. Therefore, in 1928 to 2020, Acarology unit have been conducted one study with the title Infestation of Chiggers on Small Mammals from Different Localities in Pahang, Malaysia. Abundance of the chiggers determines the possibility of acquiring scrub typhus, which in turn determines the prevalence of scrub typhus in the given region. Therefore, the objective of this study to investigate the infestation of chiggers on various species of small mammals from different localities in Pahang. The trapping have been conducted from 2018 to 2020, and then the animal have been processed to collect the chiggers, and then the chiggers have been mounting for identification based on the kits provided. In this study, we have been identified 4 genus of the chiggers, and then Leptotromydian deliense have been listed among the abundant species have been found at the Tupayag list. As a conclusion of this study, identification of the chigger species in their animal host is a fundamental for the assessment of human risk to the scrub typhus. A little bit updates on the diagnostic of scrub typhus in Malaysia. Actually, bacteriology unit is a referred center for the confirmation of scrub typhus diseases in Malaysia. This is the figure, whereas number of serum samples received by bacteriology unit in the past 3 years. And from this figure, we can see the trend of the increasing numbers of the confirmed scrub typhus disease sending from the hospital all over Malaysia. Research gap. For the research gap, there is still limited information on distribution and incidence of human scrub typhus, even though there are increasing numbers of positive cases for the past 4 years. Scrub typhus infection rates in the vector animal hosts that are in close proximity to human population are important collection sites in providing evidence-based findings to the Ministry of Health. The findings must be considered in health risk analysis and develop of control strategies, because these factors could influence human case or in combination of the ecology and environmental factors. Thus, under the Prevention of Controlled Infectious Diseases in 1988 and the latest amendment in 2011, scrub typhus is still gazetted as notified disease in Malaysia. Therefore, in 2022, Acarology unit have been approved by Ministry of Health to conduct a study with the title Detection of Orinchia Susugamushi in chiggers and small mammals in scrub typhus cases in Sabah. The objective of this study is detection of Orinchia Susugamushi in chiggers and small mammals in scrub typhus cases areas in Sabah. And the expected outcome for this study is a new complete baseline data of the occurrence of scrub typhus in chiggers and animal host species from various confirmed scrub typhus human cases localities in Sabah. The preliminary results have been identified, and then from the fieldwork at 6 localities in Sabah, and then about 225 animal hosts have been trapped, and more than 50% were infested with chiggers. This study actually still ongoing at the stage of identify of the chiggers, and then nested PCR to detect of the Orinchia Susugamushi in animal and vector. This is the proof that IMR has been listed at the world's most outstanding laboratories for study of scrub typhus. In 1948, IMR has been identified and come up with the study to control and treatment scrub typhus, and then STEM has been issued by Malaysia in 1976 as a part of three STEM issued celebrating for the Institute for Medical Research. Take-home message from this presentation. The first one, abundance of the chiggers determines the possibility of the acquiring scrub typhus, which in turn determines the prevalence of scrub typhus in given region. Then the second one, identification of chigger species related to the animal host and the presence of Orinchia Susugamushi in both are fundamental for the assessment of human risk to scrub typhus. The third one, all criteria in chiggers will incriminate they are as the true vectors and have significant medical importance because of their potential for transmitting scrub typhus to the human. The last one, we can reduce the risk to getting scrub typhus by avoiding contact with the infected chiggers by practice the proper preventive measure. Before I end my presentation, I would like to say thank you to the Director General of Ministry of Health Malaysia, Deputy Director General of Health Malaysia, Ministry of Health Malaysia, Director of IMR, the science officers, entomologists, and the beloved Acarology unit teams members in involving in my research in the chigger studies and the field works. With that, I end up my presentation and pass to you, Dr. Mariana.
Thank you, Puan Suhaili, for the insightful presentation. We will now be moving to question and answer session. Please do not hesitate to send us your questions, and I will try my level best to connect you to our speakers to answer the posted questions. Yeah. Nevertheless, should the time does not allow us to address all the questions, our speaker will try their best to answer the question through email. Okay, I got some signal here that we have received a few questions from our audience. Let me go through the questions first and pick some for you all to understand more on the ACARI that can bring significant risk to our health. Okay, the first question is for Puan Ernie. Can ticks from pets that is dogs and cats, cause tick-borne diseases?
Thank you, Dr. Mariana, for the question. Yeah, when we talk about ticks from dogs or cats, the most related genus was Rhiphicephalus species, and Rhiphicephalus-borne infection have been reported mostly in the Southeast Asian region. And if the question if can ticks from dogs and ticks transmit the tick-borne disease, the answer is yes. If the ticks are infected with any pathogenic agent such as Ehrlichia, Anaplasma or Babesia, so there is a possibility transmitting the same pathogen when they bite human. So if our pets are heavily infested with ticks, so higher chances for the owners to get bitten by ticks and then get infection. So it's such a good way actually if we do regular tick check on our pets, either dogs or ticks, and if they are heavy infestation of ticks on our pets, maybe we can see a veterinarian and ask their opinion, and probably they may consult to give you oral medication to reduce the tick infestation on our pets. That's all from me, Dr. Mariana. Back to you.
Thank you, Puan Ernie, for the answer. I hope the one that put the questions for her is satisfied with the answer. There's a lot of questions here, but I try my best to address the question to each of the speakers first. Okay. Question for Puan Suhaili. What do chiggers feed on and how the transmission of Ornithia zuzurgimushi happens? Puan Suhaili?
All right, thank you, Dr. Mariana. As I mentioned in my presentation, chiggers actually do not feed on blood. They feed on the tissue lysates or fluids in the skin cells of hosts. During the feeding time, chiggers will be pierce the skin with their sharp mouth part, and then inject the digestive enzyme to rupture all the skin cells. And then during the feeding time, the allergic reaction to the chigger saliva will be appeared on the host skins. And then with this allergic reaction actually develop one formation called stylostome, which is the hardened tube. And then during this feeding time on the hardened tube, actually the Ornithia zuzurgimushi has been transmitted between mites and the host. That's all, Doctor. Pass to you.
Thank you, Puan Suhaili. Okay, there are a lot of questions. Thank you very much for sending us your question. Question for Puan Ernie. If we are being bitten by a tick, do we need to test for disease? To you, Puan Ernie.
Okay. Thank you, Dr. Mariana, for the question. I think it is a good question actually. If we are being bitten by ticks and then later we develop tick bite symptoms such as rashes around the tick bite area, flu-like symptoms or joint pain, it is suggest if we can go to the hospital for further treatment. The doctor may suggest you the best diagnostic test that can be conducted based on the symptom that you have developed. But bear in mind that not every ticks are infected with pathogens. Thus, by removing the ticks within 24 hours, it can reduce the likelihood of the any tick-borne pathogen. Some literature says that there is no benefit actually if we do the blood testing at the time of tick bite, because the result might turn negative even though we are infected with any infection, because the infection caused by tick bite may develop later within 2 to 6 hours, 2 to 6 days after the biting by ticks. So I would suggest if you are bitten by ticks, you can observe or monitor the symptoms that you have developed, and if you have a severe symptoms such as a prolonged fever, then you go to the hospital and need to do the diagnostic test such as using ELISA or IPT. Thank you, Dr. Mariana. Back to you.
Thank you, Puan Ernie, for the answer. Next, this is quite interesting. Questions to Puan Suhaili. How can we differentiate between trigger bites with other insect bites, such as bedbug, fleas, and mosquitoes? To you, Puan Suhaili.
Okay, such an interesting question. Thank you, Dr. Mariana. Actually, chiggers, acara bites or chiggers bite have a bit different between insect bite. Normally, chiggers bites form in a line around of the wrinkled skin and then under the tight fitting clothing. And then normally chiggers bite will be will not appear any sign of biting for 20 to 48 hours. But after that, normally after 24 to 48 hours, normally the skin of the host will be appear with the red spots and the like a pimples. And then a few days after, the itchiness, severe itchiness will be feeling by the host. Compared to the bed bugs and flea bites and mosquito bites, bed bugs actually will bite at the you can get the bed bug bites at the beds, right? So normally the side of the bed bugs bites will be different, okay, compared to the chiggers, and then the symptom also will be different. Because normally the insect bite, normally it will, the swollen symptom will be appear immediately, not like a chiggers. But if the symptoms become severe, I would like to suggest to the individual or patients go get a consult from the medical provider, and then they will have a suggestion. And then maybe run a specific test. For example, if chiggers have a escas symptom, they will run the test to check either they have infected with the orange susukomoshi or not. That's all, Doctor. I pass to you.
Thank you, Puan Suhaili. Another question for Puan Ernie. From Kuantan Pahang. What will happen if the mouthparts of the tick is not completely removed? Puan Ernie?
Okay, so actually this question is the technique how to remove the ticks, right? Okay, I consider this question more to the hard ticks, which is the most common what we call family of ticks that have been discussed. So actually, removing a ticks from human body or from our animal packs is tricky, because improper tick removal may cause the mouthpart remains in the skin. So if that happen, the person might have a severe itchiness, I can say. And then if heavily scratch the area of the tick bite, it may cause unpleasant skin irritation. Based on this literature also, they said that if the mouthpart of the ticks are embedded in the human skin, it will increase the risk of to get a localized or secondary infection, and in severe cases, the person might get what we call as a skin abscession, where there are painful collection of pus and also the swollen of the area of tick bite. And if I could suggest, if the infestation of ticks on human especially occur on inside the ears or our eyes, I would suggest please go to the hospital for proper removal of ticks. They have a proper apparatus which sterile forceps or tweezer to remove the ticks and make sure we remove intact body of ticks. So there is just to reduce the any infection if we are removed by ourselves. That's all, Doctor. Back to you.
Thank you, Pak Ernie. Maybe I can add a little bit more. From my experience, most people that go inside the forest, the inner sites, especially for the armies and also the foresters or rangers, they usually mostly were bitten by the hard ticks, and it is easily to pull out if the ticks is attaching on your body sites. Okay, so normally we found on the body sites, not inside the ear canal. But for the soft ticks, one thing to differentiate the hard ticks and the soft ticks, the hard ticks, the mouth part is at the anterior of the body. But for the soft ticks, the mouth part is at the ventral of the body. And so far, I've been in service for 29 years. I have not got any soft ticks bitten. I have no experience on how the soft ticks are attached to the host. So maybe in the future we can see how we can pull the soft ticks from the animal host or from human. Okay, I think that is for the questions. Next one. Okay, I think we have a bit more time. Puan Suhaili, what is the chances to get scrub typhus if I got rodent bite with chiggers on its body? This is from Dr. Chong. Please, Puan Suhaili.
Thank you so much for the question. Actually, we have chances to get infected for the scrub typhus when we have been bitten by the infected chiggers larvae. But in these cases, actually, rodent bite actually cannot transmit the disease of the scrub typhus itself. Must be transmitted by the chiggers. But chiggers on this rodent actually half fat, meaning that if the chiggers from the rodent bite the host, and then actually the chances to get is very low because the chiggers on the rodent already fed on the host. Alright? Okay? So that's the answer, Doctor, because the chiggers actually already fed on the rodent itself. So that they might the chances be low to bite again another host. Okay, that's all, Doctor.
Okay, wow. What a good response. We can entertain another 2 questions. Okay, wait. Okay. Hope be patient because I'm screening all the questions. Try to pick up the short questions because I don't think we can reply to the long answers. Thank you very much for your responses. We are very thankful for the questions and suggestions for collaborations. Thank you very much. Okay. Okay, this one, if Puan Ernie cannot answer, Puan Suhaili should add on, okay?
All right.
What is the preventive measure can be done by the house residents that is located nearby the shrubs to prevent infestation by ticks?
What is the prevention? I think this is, I put in the slide actually, the preventive measure. One is if we have what we call, we are close to, near to the ticks habitats, maybe we can do what we call just pembersihan to at our house, and also if you notice the presence of small mammals near to your house, also you have to make sure that this animal are you have to make sure this animal not come near to your house also. And another thing is, I think the proper way to control the tick is either you use the chemical approach, or and also let's say to use the what we call as a pesticide, chemical pesticide, by spraying the area of the ticks habitats. I think that's all from me, Dr. Mariana. Maybe Puan Suhaili can add on more on the prevention.
Yeah, normally the prevention between chiggers, prevent from chiggers and tick bites, actually it is about the same, because both Acarology mostly infest on the animals and small mammals. Therefore, discourage of the rodents or small mammals at our surrounding is the best way to prevent from the exposure to the ticks or chiggers. And again, I agree with Ernie, because normally the clearing of the shrub near our houses is the best way actually to prevent from exposure to this kind of tick or chiggers mite. But normally in the rural area, they just let the house with the shrub and full with the grass. That's the place actually the most with the high distribution of the chiggers and the ticks. Okay, so maybe another thing we can, when we do sometimes at the villages people always do the farming and so on, right? So we can advise them to wear the proper attire, and then to apply any kind of repellents to prevent from the attacking by the chiggers and ticks. That's all, Doctor, from me. Pass to you.
Okay, thank you, Puan Ernie and Puan Suhaili, for the answers. There are a lot of questions, especially from the university. I'm happy to see this. Where the ticks actually for human autoacarisis, we have started from the medical officer in USM Kubang Korean, Dr. Indudaran, and I am seeing that the USM are actively doing the human autoacarisis until now. So congratulations to USM, and hope we can have a collaborations in the future. Yeah, okay. I'm afraid that is all the time we have for questions from participants. Any additional questions will be answered by speakers through email. We hope this webinar has shed light on some research work that we are conducting at our institute, and hopefully would spark interest and initiate research collaboration with others. Yeah, I can see that already in the questions, okay? So do come to us, and maybe we can discuss something for the future research. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. Please don't forget to subscribe to our National Institutes of Health Malaysia YouTube channel. And before I conclude, I would like to announce that we will continue to have the IMR-SEAMEO webinar series in the year 2023. On behalf of the webinar secretariat, I would like to thank everyone for your continuous support throughout the year in making this webinar series a success. Hope that you will be joining us again next year. Until then, Many thanks from all of us in IMR Malaysia. Merry Christmas to those celebrating and a Happy New Year 2023. Do take care and stay safe.